Good day, viewers. This is 300 Plus Academy, where all we do is exams tutoring, ensuring that you ace your next and, of course, every exam. So, the business of the day is still Neco 2024 biology practical. We've looked at all of the specimens in the previous lecture video. The link to that is going to be in the description of this video. We've looked at the very first group out of about 15 specimen. We've looked at the first group talking about onion bulb, ginger rhizome, and then Irish potato. So in this second lecture video, we'll be looking at a potted bean seedling and potted guinea corn seedling. Let's just say bean seedling and guinea corn. What are the probable questions we expect? It will interest you to know that when we talk about specimen D and E, for us to really understand and to be able to answer questions that will be posed as far as this exam is concerned, there are certain things we need to actually have a good knowledge of. One of such is, first, what is germination? These are important questions we need to ask ourselves. As you can see on your screen, you have the image of a bean seed. And from that, I'm going to be explaining certain terms like epicotyl, elum, or the scar. We're going to be looking at things like hypocotyl, the cotyledons, the radicle, and embryo, and all of that, as that is going to put us in a position to better be able to do things rightly. So, what is germination? First of all, as you can see, it is the process by which the embryo of a seed grows out of its testa. You can see in that diagram on your screen, you have a bean seed there, you have the elum, or what you call the scar, and just above that elum or scar, in the diagram just next to it, you're going to see the radicle. You're going to see the radicle, okay? So you can see that. So germination is the process by which the embryo of a seed grows out of its testa. So the green structure that you have on the inside of that bean seed is exactly what we call our embryo. So germination simply means the embryo, the green structure, growing out, you see, whereby which embryo of a seed grows out of its testa or seed coat. You can see the labeling of the seed coat there, and develops into a seedling. Now, just inside the seed coat and above the elum is the embryo, you see, just immediately above the elum, you can see it in that labeling, is the embryo, and the embryo consists of your epicotyl, your hypocotyl, and your radicle, which ultimately develops into the root. Look at it. I try to capture all of that written on the board. The tip of the embryo is called epicotyl, and the epicotyl is going to become the meristem of the shoot. Is going to be what is going to develop into the shoot, carrying the foliage, leaves, and the cotyledons. Look at it very well. I said epicotyl. The tip of the embryo is called epicotyl. Look at the inner structure of that bean seed here. You can see the epicotyl is at the tip. Even in other diagrams, as you can see for maize seedling, you can see the labeling of the epicotyl. It is the tip of the embryo. So the short greenish structure you have on the inside of the bean seed is the embryo. So the tip of that embryo is called epicotyl. The radicle, and that epicotyl is what is going to, carry the shoots and the foliage leaves. The radicle is going to develop downwards, and that is what is going to form the root. Now, in between, and that's exactly what I captured there. I said the tip of the embryo is called epicotyl. The epicotyl becomes the meristem of the shoot. Now, the embryo is the radicle, which will develop into the root. So you expect the radicle, as you can see in that diagram, to go downward and develop into the roots. Now, the epicotyle and radicle are connected by a tissue called hypocotyle. You can see that, hypocotyle. Now, if you look at that particular diagram, you see on the inside of that bean seed, the hypocotyle is in between the epicotyle and the radicle. And if you look at the normal germinating seed here, if you look at it, the hypocotyle is between the, you can see, hypocotyle is between the epicotyle, which is holding the leaves, is the shoot, epicotyle, and the radicle, which forms the root. So in between the root and the shoot is where you now have your hypocotyle. Now, connected to the embryo are two leaf-like structures called cotyledons. You can see the cotyledons on the side. You see, connected to the embryo are two leaf-like structures called cotyledons. Now, look at, in a maize seed, in a corn seed here, we have guinea corn. In a corn seed here, you can see where you have your cotyledon. So your cotyledons absorb food molecules from surrounding endosperms. You can see the labeling of the endosperm and transport the food to the embryo, where you have the epicotyle. You see where the cotyledon is. So that is what is going to absorb food from the endosperm and transport that to the epicotyle, hypocotyle and radicle, which all together forms the embryo. So that's just about something basic that we need to note, so that as we continue to discuss, it's something you can get along with very well. So what are the expected questions for specimens D and E? As you can see on your screen, the first question states that states the germination in D and E with reasons. So what is expected here is you're to observe specimen D, the potted bean seedling, and you're to observe specimen E, the corn seedling. Well, in D, as you can see also on your screen, the germination is actually epigynous germination. And why am I saying epigynous germination? Because the question says with reasons. First, the cotyledons, as you can see in the diagram on your screen, appeared above the root or the soil, and also the hypocotyle is elongated. The hypocotyle, you can see it before the cotyledons, between the radicle, that's the root, and the cotyledons, before we then talk of the epicotyle. You understand? So the hypocotyle is elongated. That is why I am classifying the bean seedling as a type of epigynous germination. When you talk about specimen E, you can see that that is hypogynous germination, simply because the cotyledon remained below or beneath the soil. And in this case, what we have is just the epicotyle. The whole of the epicotyle appeared above the soil. You cannot even see in the labeling there, that's above the soil. You cannot see your hypocotyle there. So we just say epicotyle appear above the soil. That is why we are classifying the germination in specimen E as hypogynous germination, and that ends that question. So moving on to the next question, we're told to describe the germination in D and E. Don't forget, the germination in D is epigynous germination, and the germination in E is hypogynous germination. So the germination in D, we were told to describe that. You can see the diagram on your screen. Seed absorbs or imbibe water and soil. The seed is going to be in the soil. So the first thing that happens is that it absorbs water and it swells. The testa splits and the radicle emerges. You can see how the radicle, which is eventually going to form the root, you can see how it emerges in that diagram. Hypocotyle elongates, pushing out the seed from the soil. You see, hypocotyle elongates, pushing out the seed from the soil. Recall that the tip of the embryo is the epicotyle. So I've said in between the radicle and the epicotyle. That is where you have the hypocotyle. So the hypocotyle here elongates, pushing out the seed from the soil. Now the cotyledons now are exposed to sunlight, and eventually they turn green to be able to photosynthesize. Now the plumule develops into a green shoot. By saying a green shoot here, it develops into a foliage leaf. Develops into a foliage leaf. It develops into a foliage leaf, and with time the cotyledons dry up and they shrink and fall off. So that is just what epigynous germination indeed entails. Look at the diagram very well. You're going to see the cotyledons, the way they are being pushed out. You see the tip, there is the epicotyle. In between the epicotyle and the radicle is where you have the hypocotyle. So for this epigynous germination, the hypocotyle elongates, pushing out the seed, and all of that, so that just about ends the germination we have in specimen D, epigynous germination. You can see that diagram clearly on your screen. So still on question 2, let's quickly look at the germination in specimen E and describe it. So talking about the germination in specimen E, it is actually hypogynous germination. So what happens here, it is always about the seed. It absorbs water, or it imbibes water and soil. The seed coat or tester breaks because why? The radicle has to emerge and it grows downward to form the root. In this case, it forms a taproot system. Of course, the epicotyle grows and loops above the soil, pushing out the plumule. The epicotyle will then straighten. The plumule, in this case, as you can see in the diagram on your screen, the plumule is going to turn green in the presence of sunlight to photosynthesize, and that's going to form the first foliage leaves. Look at the diagram that you have on your screen. You can see what we mean by the foliage leaf. You can see the epicotyle as it straightens up. You can see the emergence of the epicotyle. You can see the straightening of the epicotyle. You can see the radicle growing downwards, and you can see the formation of the first foliage leaf. There you can see the taproot system there. You can see the primary root, and then you can see as well the tiny branches, which we call the secondary root. So it's a very direct explanation here. Epicotyle grows and loops above the soil, pushing out the plumule. Epicotyle is then going to straighten. The plumule, in this case, you can see what we mean by the plumule. You see? You can see what we mean there by the plumule. So epicotyle is going to straighten up, and the plumule is going to form the first foliage leaves. Radicle is going to develop into a taproot system. Here we don't talk of hypocotyle, and you can see that the cotyledons remains beneath the soil. So that is germination in E. Moving on to question 3, it states the conditions for germination. That's we should state the conditions for germination. Now, the condition needed for germination, you can see here that water, seed absorbs and imbibe water. So the very first condition is water, or what we call moisture, and then we talk of warmth or suitable temperature here. Then we talk of air, and then the seeds, in this case, so that germination takes place, has to be good seed, has to be viable seed. So these are the conditions for germination of a seed, and that ends question 3. So moving on to question 4, we were told to differentiate between the germination in D and the germination in specimen E. As you can see on your screen, the germination in E is actually epigynous germination, and the germination in E is hypogynous germination. D is epigynous, E is hypogynous. So what are the differences? You can see that in epigynous germination, the hypocotyle elongates. We've said that before in the description, but for hypogynous, it is the epicotyle that elongates here. The cotyledon, in the case of epigynous, emerges, grows above the soil. The cotyledons in the case of hypogeo germination, in this case of specimen E, remains in the soil. Now, in epigeo germination, number 3, since the cotyledons emerges, it turns green and also photosynthesizes. No wonder you see that in the diagram, the cotyledon in the case of epigeo germination for specimen D is painted as green. But in the case of hypogeo germination, cotyledons remain in the soil, so they do not photosynthesize. For difference number 4, the plumule does not emerge at the same time as the radicle. You can see, look at the diagram very well. When do we begin to talk about the plumule? The first thing that develops here in the case of epigeo germination is the radicle. So it is later on that you're going to talk about the plumule. You can see that in the diagram. The radicle develops, but the plumule does not emerge at that time. Later on, that is when the plumule is going to develop. So when you look at epigeo germination, plumule does not emerge simultaneously with the radicle, but in the case of hypogeo germination, plumule emerges simultaneously with the radicle. Also, the energy and the radicle itself here comes from the cotyledons. Yes, the energy needed here is derived from the cotyledon, and when you look at hypogeo germination, the energy is derived from the endosperm. Yes, the energy is derived from the endosperm. We talked about it at the start. You can see where the endosperm is here. So the energy that is needed here is going to come from the endosperm, and those are just the major differences between the germination in specimen D and the germination in specimen E. So moving on to the next question, talking about question 4A. As you know, biology practicals are not complete and can never be complete without a drawing. So in question 4A, we were told to make a drawing here. The question says, make a drawing 8 to 10 centimeter long of specimen D. Yes, make a drawing 8 to 10 centimeter long of specimen D and label fully. So it's about time we get our pencil and we try to draw things. So let's see what things should look like. I think when we are starting, okay, let's start from the top. Yes, let's start from the top. Starting from the top, when they say 8 to 10 centimeter, okay, let's say this is my 0 centimeter mark, and let's say this is my 8 centimeter mark, and let's say this is my 10 centimeter mark. Now, what this means is that my drawing should not be less than 8 and should not be more than 10. So the drawing should start somewhere around there and should, fall within here. Should not be more than 10 centimeters. So what is the starting? Okay, just in case we were told to draw. Okay, I'm going to, have this line. Then I'm going to have this line. Then I'm going to create a space, a space so that I can use that for my foliage leaves. And then I'm going to have this line as well. Kept on going on. Then I'm going to have another space because this time around I want to talk about my cotyledon. Okay, I'm going to have space here as well because I want to talk about my cotyledons. So that way I continue downward, so you can see I started from 0. I'm nearing towards 8 now. I've given space, for my foliage leaves here. So I could go like, okay, let me make this a bit more close so that if I say this is my leaf here, I could go like, okay, I could go like, okay, and then I could go like, these are my foliage leaves. I could draw the veins, I could draw the veins for my leaf. So this is my foliage leaf on this side, and then this is my foliage leaf on this side as well. You see that? See that? So I've been able to do that, and then I can go with my veins, you know. Don't want this to come out. I can go with my veins, you know, just something we can actually flow with. Simple enough. So I've been able to do that. I've been able to draw my cotyledon. What else do I need to do? I just about now I need to begin to move and descend into the root system, and I don't want that to be more than this. So first off, I want to draw a single taproot system. You see that? I had to curve it because I don't want it to go outside of the 10. So now that I've done this, then I can begin to talk about, you know, the various branches of the taproot system. Okay, this is going to represent our cotyledons here. This can then begin to give off branches, you know. This can then begin to give off branches. This can then, numerous branches, by the way. You know, just do your thing. It must not be more than the 10. It must not be more than the 10. You know, just numerous branches from the root system here. Yes. So when you look at this, this just does it. So we can now talk of actually getting this label. This is going to be, of course, this is going to be, you know, the level of the soil. This is going to be the first foliage leaf. Okay, and what we have in here is the epicotyle. Now, in between the epicotyle and the radicle, that's talking about the root, is where you have your hypocotyle. And immediately after that, you then have the soil, and then you have the main roots. Yes, main roots. I can take this off now. You have the main roots. You can see what I'm trying to do. The lines just matches. You have the main roots, and by the side, what you have are what you can call the lateral roots. But most importantly, these are the important parts. And here you have your cotyledon. So I can take off my 0 centimeter. This you're going to label as drawing of specimen D. Specimen D is being seedling. So, and that just about ends that. That's talking about 4A. We were told to make a drawing 8 to 10 centimeter long of specimen D. That's the diagram there. So the next question tells us that's 4B. It says we should state one function each of any 4 of the labeled parts. We should state one function of 4 of the labeled parts. We should state one function each of 4 of the labeled parts. Let's look at the foliage leaf. Talking about the leaf, the function of the leaf for a plant is to manufacture food via photosynthesis. So foliage leaf, it is to manufacture food via photosynthesis. Okay, talking about the epicotyle, epicotyl and hypocotyl. Their function is very simple. It is to hold the foliage leaf above the ground so that the leaf receives sunlight. It is just to support the leaf, to support the cotyledons, which is actually the next one. Is going to provide food for the developing seedling. You can see that it's going to provide food from the developing for the developing seedling. So it's a very direct thing. Epicotyl, hypocotyl, they hold the leaf above the ground so that it receives maximum illumination or sunlight. That way, photosynthesis or manufacture of food is able to happen. And then the function of the cotyledon is that it provides food for developing seedling. Okay, let's take a look at another part. That's the main root now. The function of the main root is water and mineral salt absorption. So moving on to the next question, we were told to give the agricultural classification of specimen D and specimen E. Specimen D, talking about the bean seedling, is a legume, and specimen E is a cereal. Don't forget that D is a legume, E is a cereal. And to the next question, we were told to name the part where food is stored in specimen D and E. D is bean seedling, E is a corn seedling. Where exactly is a food being stored? In the case of D, as you can see in the diagram on your screen, food is stored in the cotyledon. Food is stored in the cotyledon. And in the case of E, food is stored in the endosperm. You can also see that diagram on your screen. So in the case of E, because it is hypogeo germination, you see that the cotyledon in E is actually going to absorb food from the endosperm where food is being stored, so that the food can then reach the embryo. Okay, that ends that. So food storage in D and E. Food is stored in the cotyledon for D, and food is stored in the endosperm for E, talking about the corn or maize seedling. Now, to the next question, as you can see on your screen, we were told to state the economic importance of specimen D, talking about the bean here, the bean seed here. We were told to state the economic importance. It's a source of protein. We all know that. And also, it provides a cover crop, preventing soil erosion. It provides what? Cover crop preventing erosion. It is also used in crop rotation for maintaining soil fertility, so that the soil is fertile. Specimen D is a plant that can be used in crop rotation. It can be used as cover crop to prevent erosion. And also, talking about the roots, the root nodules harbor bacteria. It harbors nitrifying bacteria, talking about Nitrosomonas, Nitrobacter, which helps to convert atmospheric nitrogen to nitrates, thereby making the soil a lot more fertile. Remember that nitrates are one of the macronutrients that we need to have a luxuriant plant. And that just about ends that for specimen D and E. And with that, we have come to the end of the second lecture video, talking about Neco 2024 practical. For myself and the entire team, it's bye for now.